Hi, Mauro.
Hi, how are you?
Good, good.
Good.
So I have the record ready to get started. Yeah, it's 5 after the hour. So welcome everyone to the Friday Physics Colloquium. So our today's speaker is Dr. Mauro Tambasco, who's currently at San Diego State University. He started off, got his undergraduate at University of Toronto, a PhD in medical biophysics at what's now known as Western University in the state of Ontario. He did a 2-year residency in radiation oncology physics at the University of Toronto. And he was a professor for 8 years at University of Calgary and a medical physicist in the Department of Radiation Oncology. And after that, he became a professor at San Diego State University, which is where he currently holds his position. So today's talk is going to be on assessing DNA damage in radiotherapy applications using a DNA-based detector. So Mauro, the floor is yours.
Great. Okay, thank you. All right. Everybody can see this?
Mm-hmm.
Great. All right, thank you for the invitation to speak here. I was supposed to speak at Fresno a couple of years ago. I think I was delayed with the plane, but hopefully now nothing goes wrong. All right, a few developments since then, but some of the talk is the same as what I was going to give. So yeah, it's assessing DNA damage in radiotherapy applications using a DNA-based detector. So now most radiation therapy is done these days. It's external beam, and it's using Linacs, like these the machine shown here in the figure, and it's mega voltage energies, and it's X-rays. And these machines have image guided systems. Right now, most of them are X-ray based, so it can do like cone beam CT to get like X-ray images. And the patient would lie on the couch here, and the beam would come out of the portal here and treat the patients from different angles. However, recently there's been a rise in proton therapy in the United States. As you can see here, all the red dots popping up. And in other parts of the world, there's also been a rise in another heavy particle these are charged particle beams. So another heavy charged particle beam, the carbon ion beam. And so we have carbon ion radiotherapy, or CERT. There's about 50,000 patients that have been treated worldwide. 8 operational centers. There's 5 in Japan, 1 in Germany, 1 in Italy and Austria. And there's centers in South Korea, Taiwan, and China that are going to open soon. There are none in the United States. However, there are several in development. Now, the reason I mentioned these is because they have different relative biological effectiveness, and I'm going to define this in a minute, than the radiation therapy that has been traditionally used, the mega voltage X-rays, that first slide that I showed you. So if you have equal doses of different types of radiation, they do not necessarily produce equal biological effect. So for example, 1 gray of carbon ions produces more biological effect or greater biological damage than 1 gray of X-rays. And to define the effectiveness, or the biological effectiveness, there's this term here, it's RBE, it stands for relative biological effectiveness, to compare these different radiation types. And the definition is, it's the dose of the reference radiation to give a certain biological effect over the dose of the test radiation to give the same biological effect. Now, the reference radiation traditionally was taken to be the 250 kVp X-rays or the cobalt-60 gamma rays. But now, these days, when RBE is calculated, since these X-rays are not very common in radiotherapy, what's used is the 6 MV X-rays, these 6 megavoltage X-rays as the reference dose. So that would be the numerator here. And then in the denominator, you put the dose that gives the same biological effect from your test radiation. And that could be the protons or the carbon ions or whatever your other radiation is. So here's just a diagram. If you consider an experiment where you're irradiating like human cells and you irradiate them to different doses, you can construct curves that look like this, which plot the surviving fraction as a function of dose. And we see that for different radiation types, we get different cell kills. So if we pick an endpoint, for example, 10% survival or 10 to the minus 1 surviving fraction, we see that we need a much higher dose of X-rays to reach, only 10% of the cells surviving than we would have for, say, 15 mega electron volt neutrons or for alpha particles. But like was said earlier, before the talk, alpha particles, you could stop them with a piece of paper. So they're not really used in radiation therapy, but this is just to bring the point across that different types of radiation beams will give different biological effectiveness, will have different effect. And so if you give the same dose, you're not going to have the same effect. So you pick a constant dose here, you will see that different levels of survival will happen, right? Now, for the beams I've mentioned earlier that are used in radiation therapy, the first was the megavoltage X-ray beam, as shown in the green line here. Now, this graph here shows the relative dose effect as a function, or the dose as a function of depth. So how much dose you're depositing as a function of depth into the body. And for megavoltage X-ray beams, they typically have a curve that looks like this. It's low dose near the skin or the surface. And as you go deeper, the dose deposited increases and then starts to decrease again. But for proton beams or carbon ion beams, you can modulate these types of heavy particle beams so that they also have a relatively low entrance dose and they stay low, and then they rise, and you can get them to rise wherever you want. For example, if the cancer is between the depth of roughly, just over 6 to over 10 centimeters here, you can get it to rise and plateau, so you deliver a uniform dose to your tumor. And then on the other side, they drop really rapidly. So you get almost, zero dose here. So if you have a critical structure here that you don't want to receive any dose, it's good for that. And it's good for giving, relatively low dose near the entrance. Whereas the X-ray beam doesn't have this kind of control. You can't get this kind of control with X-rays. And the way you get this, what we call spread out Bragg peak, this flat region with the carbon ions and the protons, is you modulate the energy of the beam. Because if you just have one energy, what you would get is called a Bragg peak type of curve, which has a very low entrance dose. And then at some point, it really peaks really steep and then drops really fast. But you can modulate it, and because tumors have a wider region, and to get that region, you need to modulate the energy, and so you can get this flat dose response like here. Now, both, you can see from this diagram, both the carbon beam and the proton beam, these heavy charged particles, offer a dosimetric advantage over X-rays because you can avoid normal tissue dose distal to the target, and also proximal to the target, you can get less dose. Sorry about that. And you have lower entrance dose here. So, but with protons, we have a relative biological effectiveness that varies over this spread out bright peak. And it's about 1.1 to 1.3 times that of the megavoltage photons. And it can be higher as you go across here from one end to the other. And right now, clinically, there is no way to measure this relative biological effect in an easy way, because clinical medical physicists, when they're looking at the treatment plans before a patient is treated, they will do measurements with physical dosimeters that measure, like ion chambers or diodes or thermoluminescent dosimeters or film. They use these kind of dosimeters, but they measure radiation dose, which is the amount of energy deposited per unit mass at a particular point. But we can see here that the relative biological effect for the same dose can be different. So dose doesn't tell you how much damage you're going to cause. And right now, physicists don't have a detector to measure this. And this is the same thing happens with the carbon ion radiotherapy, where you have a radiobiological effect. And in this case, it's even bigger than with the proton beam compared to the reference radiation, which is the megavoltage photons. And again, right now, we have no way of measuring this RBE effect. And just to understand why we get this difference in radiobiological effectiveness, I have just this schematic diagram, and it shows the two types of actions that ionizing radiation has on damaging DNA. And by the way, DNA is the target that causes your cells to die, because if you damage the DNA irreparably, you'll cause enough cell damage that the cell will just kill itself, commit cell suicide during its division or mitosis. And so what happens, you have the indirect damage. Now, ionizing radiations like the X-rays, they tend to do a lot of this kind of indirect damage. So they come in, they ionize the water molecules that are around the DNA molecule, and this results ultimately in free radicals that are close enough that can diffuse to the DNA molecule, and they can cause a strand break to the DNA. Alternatively, you can have this direct action where the ionizing radiation hits the DNA molecule directly and causes strand breaks. And with the heavy charged particle beams, you tend to have more of this direct action versus this indirect action. And this ends up leading to more cell kill, because you'll end up getting more double strand breaks with this direct action. And it happens with these heavy charged particle beams. So just to see that schematically, if you had a carbon ion beam traveling through some material here, as shown, you get by this dashed red line, you would get a lot higher density of electron ionization or electron tracks shown here. And these electron tracks cause a lot more ionizations than you would get from the proton beam, again shown by this red line. So if a proton beam or proton is traveling along the red line, you get much more sparse ionizations. So if you have a DNA molecule along the path, you are more likely to hit it with a high density of ionizations that you would get with the carbon ion beam than with the proton beam. And that means you would get a higher relative biological effectiveness with this heavy charged particle than this one. And of course, with the proton beam would give you more damage than you would get with the same dose of an X-ray beam. Now, I'm just going to switch gears for a second and talk about another technology that has been emerging in radiotherapy, and that is magnetic resonance image guided radiotherapy. So I showed in that first diagram that we have imaging systems attached to the linear accelerators that deliver the radiation beam. But most of these imaging systems have been using X-rays, doing CT or cone beam CT scans, which are great. They give you 3D images, but the soft tissue contrast with CT is not very good. But with magnetic resonance imaging, you really have good soft tissue contrast. And this makes it advantageous when you're doing radiation therapy because the tumors are soft tissue. And if you want to be able to see the difference between the tumor and the background normal tissue during treatment, before treatment, or even after treatment, you can do it better with an MRI machine than a CT. So they've integrated the MRI, as you should see in this diagram here, and here you can't really see it. This is another system. This uses cobalt sources, and this one uses a linear accelerator. And the reason it took so long to get these machines is because MRI machines are not so compatible with the Linacs because the static magnetic field of an MRI machine will interfere with the electromagnetic components of the linear accelerator structure of a Linac. But the engineers and physicists got this to work. So now these are actual clinical machines. There's not that many of them out there right now, but they're gaining ground again because they can give you that soft tissue contrast that you can use to see the tumor during, before, or after the treatment. And they've also developed MRI guided high dose rate brachytherapy. And this is, brachytherapy is when you have a radioactive isotope that's inserted into a cavity, into a patient. It's for HDR, it's iridium-192, and it's inserted through a cable and it dwells inside the patient and then it's pulled back out by the machine. And again, you can have an MR guidance to see where that source is going and to see the soft tissues during the treatment and make sure that that you're delivering the dose as you want to. And they've also talked about implementing magnetic resonance imaging for proton therapy, although there's no existing machine to do this just yet. However, there's been some theoretical work by physicist Dr. Pang that showed that the static magnetic field of an MRI machine could alter the radiobiological effectiveness of your linear accelerator X-ray beam. And to understand how it does this, in the presence of a strong magnetic field, you'll have this Lorentz force. And if you have an electron with some velocity, it's going to then result in this force inward. So that electron, instead of traveling as it would in a straight direction, it'll travel in this kind of spiral direction. And the reason this was postulated to possibly increase the radiobiological effectiveness is because if an electron was to cause an ionization at point P1 here, and then a second one at point P2, so if the effective length or expectation value of the path length between ionizations of an electron is given by, say, this distance between P1 and P2 prime. But now you introduce a magnetic field which curves the electron's path. This arc length is the same as the length between P1 and P2 prime. However, the chord, which is the straightest distance between these 2 points, is shorter. And so you can see that the number of ionizations, or the ionizations distance is shortened between these 2 points. And so it's like you have a more densely ionizing track, if you imagine a whole bunch of this happening. And if the track gets more densely ionizing, and another definition to characterize this is linear energy transfer, which is the amount of energy transferred to the medium through these ionizations per unit length that the electron travels. If that increases, then you have a higher relative biological effect, and this results in more double-strand DNA breaks. And it's these double-strand breaks in DNA that can be lethal for the cells. These are the breaks that can kill the cells and cause them to die, by definition, if you kill them. Another way to see this is if you have no magnetic field, and here's a DNA molecule, these dots representing ionizations by the electron tracks, you get fewer of them, so there's less ionizations, less damage. In the presence of a magnetic field, if the distance between these ionizations is shortened, you could potentially get more ionizations causing more DNA damage, as shown here. And also it's been postulated that in addition to shortening the track, there could be radiochemical effects. So for example, the static magnetic field may influence the yield or lifetime of the reactive oxygen species that are responsible for the indirect DNA damage. And this could be a different radiochemical effect that can also increase the amount of DNA damage in the presence of a strong static magnetic field. So some of the research goals I've recently focused on was to test this. So we constructed a DNA-based detector that can discern changes in radiobiological damage due to altered the nanodosimetry and or radiochemistry. And we use this detector to characterize the possible changes in DNA damage and its associated relative biological effectiveness. And we're measuring relative biological effectiveness here in terms of DNA double-strand breaks. And we did this in the presence of a strong static magnetic field and for modulated proton radiotherapy beams. So the basic idea behind a DNA-based detector is that the DNA damage is the principal contributor to cell death by ionizing radiation. Hence, a DNA-based detector it lends itself naturally as a tool to measure the direct damage of this radiobiological damage. So it's a direct measure, unlike an ionization chamber where you're not measuring damage to a biological molecule, but you're just measuring the amount of ionizations created in your detector. So a DNA-based detector can be used to measure the yields of single-strand breaks, or SSBSs, and double-strand breaks, DSBs, in DNA due to ionizing radiation. And this direct measure of DNA damage will allow for computing the relative biological effectiveness, with the endpoint being the DNA double-strand breaks. So here's just a picture of the detector design that we made. This is one of them. We used a polycarbonate material because it's hydrophobic and biocompatible, and it's nearly tissue equivalent, meaning it has a density and effective atomic number close to that of water, which is, just like saying close to that of tissue. And we basically drilled 9 cavities in this material with a depth of 2 millimeters and a diameter of 2 millimeters. So they're cylindrical cavities, giving a total volume of 6.28 millimeters cubed, or microliters here. And we placed 3 microliter samples of DNA solution into each of these cavities. And then we seal it with an aluminum seal to prevent evaporation of the DNA. So once we've done this, we now have our detector, which we can then place in the different types of radiation beams, and afterward take the aluminum seal off and analyze the amount of damage done to the DNA. Now, the DNA, I should have said here, that is put into these detectors is 90% supercoiled plasmid DNA. And we bought this from Thermo Fisher Scientific. This DNA has 4,361 base pairs, and we place it inside our little cavities. Now, the reason why we use this plasmid DNA is that it comes in this supercoiled form or conformation, as we see here. And you can imagine this as if you take a rubber band and you twist that rubber band, you twist it up into a really small form, then you get this kind of all twisted up supercoiled conformation. Now, if the radiation comes in and causes a single strand break to the DNA here, this rubber band would uncoil and form a circle. So the DNA then forms this conformation, the open circular conformation, if you get one single strand break. If then you get another single strand break here, it breaks this molecule apart so that the conformation turns into a linear form, as shown here. Also, you can get a linear form by a direct double strand break, going from supercoiled. If you break both strands here, you'll go directly to this linear conformation. And it's this double strand break or this linear conformation that can be deadly to a cell. And it's what we want to measure. So now we want to measure how much, if we expose the supercoiled plasmid DNA to radiation, how much of this linear conformation do we get? And before we go further, I just want to mention that DNA has a charge, the reason is it's got these phosphate phosphate groups on the backbone of the DNA molecules. So these are the strands here. And in between, you have the rungs of the DNA ladder made up of the base pairs. And these are negatively charged. And so we could take advantage of this to separate these three conformations. They all have they have the same charge, but because they're different forms, they're going to travel differently if we put them in a gel. So a common technique in biology is to use Agarose to do Agarose Gel Electrophoresis. So since DNA is negatively charged, if we irradiate our samples and then we place them in a device like this, which has two electrodes, a negative electrode and a positive electrode, and we seed them in a gel, an agarose gel, they will travel from the negative electrode side, since they're negatively charged, they'll be pulled toward the positive electrode. And those different conformations of the DNA will be pulled through the gel at different rates, because the agarose gel will have pores, and that electric field will pull the DNA, but the supercoiled, it turns out, will slither through those pores a lot faster than the open circular DNA, and then the linear DNA will be somewhere in between. So if we set our device here to an electric potential of 100 volts, we apply 100 milliamps, and for 80 minutes, and we stain our DNA so that we can see it later, the negatively charged DNA will get pulled across, and they'll travel different distances because of their different conformations. And then we can later bring that gel, put it under a blue light transilluminator, and take an image of it with a 14-bit grayscale camera, and then measure the intensity of the bands that we get, and in that way quantify how much DNA had no strand breaks, how much had single strand breaks, and how much had double strand breaks. And this is just a schematic showing that. So we would get 3 bands like this in our gel when we image it. And the top band will correspond to the open circular because this travels the slowest. It has the hardest time slithering through the gel, going from one end to the other. The linear will go a little further, and this corresponds to those double strand breaks. The circular was the single strand breaks. And if we had no damage, that would be the supercoiled, and it travels the fastest. And then we can quantify the intensity of these bands again to see, how much double strand breaks we got when we exposed the DNA to these different types of particle beams. So here's an actual image of a gel we ran. And the DNA is migrating from the negative side here to the positive. And in the control, we see that most of it is supercoiled. So when we buy this DNA, it's not 100% in the supercoiled form. 90% of it comes in the supercoiled form, and 10% is in the open circular form. And we see that here. So we just see 2 bands. And then if we irradiate it, suppose we irradiate to 20 gray, we see here a lot of that supercoiled band has now disappeared. So the signal here has disappeared because we caused a single strand break. So the supercoiled DNA transformed into this, this type of conformation, the open circular. So this lights up a lot more. It shows you've caused some radiation damage. And we also have a faint linear band here showing how that we've also gotten some double strand breaks. Now, if we go even higher to 30 gray, the open circular band gets even brighter, and the linear band as well gets brighter, and the supercoiled band gets fainter because we have less supercoiled, because the supercoiled is your source of these other 2 conformations. So when it gets irradiated, this supercoiled will change into one of these ones. And then we quantify these bands, and that's, again, how we measure the amount of single and double strand breaks we've caused with the radiation beam. And this is done, we just measure the pixel intensities. We actually take the intensity of every pixel and we sum it up, and we subtract the background of the image, so that we just get the band intensity. And then, in addition, we have models, published models there for how the plasmid DNA transforms from the supercoiled to open circular and to linear. So we can, and how it transforms as a function of dose. And also we have, these parameters telling us how much supercoiled we started with. So if it's 90%, S naught represents 90%. We started with supercoiled. C naught means some of it wasn't all supercoiled. So 10% of it was open circular. So we plug in our parameters. We plug in the doses. We solve this system of equations, and we need at least 1 dose to do so. It's an overdetermined system, so we use an evolutionary model by minimizing the chi-squared. And we solve this system to get the different conformations as a function of dose. So if we, for example, if we did this experiment with these 3 possibilities, 0 dose, 20, and 30, we have all the information we need now to know what the yields will be of supercoiled, open circular, and linear DNA as a function of dose. And then we can plot curves like this, showing how, as expected, the supercoiled, and it turned out for this particular run, we had less supercoiled at 0 dose. It was actually less than 90%. It was around 75%. And it decreased as you go increase in dose. And we see the open circular or single strand breaks increase really rapidly and then kind of plateau. And the the linear band, or the double strand breaks, increases linearly with dose. And in this experiment, we actually compared, a static magnetic field with a no magnetic field. And we see that as you increase the dose in the presence versus no magnetic field, it turns out there's no difference in the DNA damage that was theoretically predicted to possibly be larger if you irradiate in the presence of a strong magnetic field. Optimization of the method. The sensitivity of the method will depend on the following factors: the concentration of the supercoiled DNA in the solution, the concentration of the agarose to form the gel, the electrophoresis settings, the fluorescent stain used to image the DNA, the scavenging capacity of the solution sample. This scavenging capacity, what this means is there are free radical scavengers in the DNA samples that can, when free radicals are formed from the radiation, they can neutralize them and prevent them from damaging the DNA through the indirect damage method. The temperature of DNA during irradiation. If you irradiate the DNA at higher temperatures, you're going to end up getting more damage. So we optimized a lot of these parameters and tried to keep the temperature constant when we did these experiments. And you can use the sensitivity analysis data gathered to fix the parameters. So the DNA-based detector is sensitive enough to detect damage at clinically relevant radiation doses. So those parameters I talked about on the previous slide, we played around until we were able to make our detector as sensitive as possible to the doses that we wanted to detect, and also to minimize the measurement and analysis time and minimize the measurement uncertainties. So for our first experiment, we used this unit here. It's called a high dose rate brachytherapy unit. This is the HDR unit, high dose rate brachytherapy unit, that I was talking about early on in the lecture here, or the talk here, where the iridium-192 source comes out of this cable, travels into a patient, if you're treating a patient, and it's programmed to different dwell times to treat the tumor to a certain dose. And we use this to expose our DNA. And then the reason we use this is because we wanted to also have a strong magnetic field in place. And we achieved that by using a bipolar magnet, as shown here. So we created a, we call it a phantom in medical physics. It's just an object that mimics the tissue properties of a patient. And we put it between the poles of the magnetic field, and we had the source here come out and go into this region between the strong magnetic field. Magnetic field was set to 1.5 Tesla. And to achieve that, we had to have a pole separation of 1 centimeter. And this was the actual phantom. We had to modify the phantom for this use. So we had the phantom, it looks like this. It was 10 centimeters wide and I think 15 centimeters in length here, and about 8 millimeters in width, as shown here in these schematic diagrams. And so we placed our DNA solution into a cavity here, and we had the radioactive source come through either this channel, this hole here, or this channel here. And you see the setup here. We had the magnetic field. These are just ice packs to keep because the magnet would heat up, the coils would heat up over the delivery of the dose, so to keep it cool. And you see the catheter here, or the transfer tube, going into one of these channels to come in here, and it would dwell in there to give a uniform dose to the DNA sample here. And this just shows the cavity dimensions, the cross section here. So this is a top view looking at it from the top, and this is a side view. And you can see the DNA was in here, and it's 4 millimeters deep this way and 2 millimeters across this way. And to do the delivery, we had to scan that block, this Phantom. We had to scan it in CT, get the proper density, and then we could make a treatment plan. This calculates the dose before you actually deliver it, so it can determine how long the iridium-192 has to dwell in those, cylindrical cavities here that we drilled for the source, so that you give a uniform dose to the DNA here. And we see that we get a uniform dose within plus or minus 3%. So we also used radiochromic film in that Phantom. We put this radiochromic film, it was EBT3 gaphchromic film to be more precise, to make sure that the dose we delivered was in the presence or absence of a magnetic field was the same. So that, in other words, we're delivering the same dose, but we're getting a different relative biological effect because the presence of the magnetic field could alter the relative biological effectiveness, even though the dose would not be changed. And we see here, in fact, what the film showed us is that the dose was the same, statistically the same anyway, between the 0 and 1.5 Tesla field. There's no meaningful difference between them within the uncertainties of our film measurements. And so any changes we do see then would be because of the relative biological differences between having a magnetic field present during the radiation and not having it there. So here's just a sample of the gel showing, 0 dose and in the absence and presence of a magnetic field, just to see if the magnetic field alone made any difference to single or double strand break damage. So the question was, will a magnetic field by itself damage DNA? Then when we delivered 20 gray, in the absence or presence of magnetic field, what is the difference in damage? And also with 30 gray. And so we had 30 controls. So these were the control, the 0 gray, and we had 13 measurements at 20 gray and at 30 gray. So we had enough samples here to have a statistically meaningful result to see if there's any difference here between having a magnetic field or no magnetic field when we deliver this radiation. And what we found is no statistically significant difference in the double strand break damage. And remember, this is the damage that can be lethal to cells, this double strand DNA break damage. So we found that the presence of a 1.5 Tesla static magnetic field that you would get from the magnetic resonance imaging present during irradiation did not cause any extra damage in the DNA. And this is, these bar plots showing, again, the 3 different groups. And the middle one here, that's the double strand break damage. So here we have it for the control, the magnetic field alone, 0 radiation. And there was no statistically meaningful difference there, nor was there at 20 gray or 30 gray. So we answered that question, and because theoretically it was postulated maybe there will be a difference and we have to account for it because we're treating patients now with a new technology in which we have a strong magnetic field, and we really want to know, is there going to be a difference? So our DNA-based detector was a way to answer that. Now going back to proton therapy, we also used the DNA-based detector to measure DNA damage of proton compared to photon radiotherapy. So delivering the same dose and to see if we detect any variation in DNA damage across that spread out Bragg peak that I talked about earlier. So again, a proton beam, if you have just a single monoenergetic proton beam, the amount of dose it deposits as a function of depth is low at the entrance. And as it goes deeper and deeper, that dose goes really high and then drops off really rapidly. And that's a good property for a beam, especially if you have a really tiny tumor, which is not normally the case. So normally you don't use a proton beam just with a mono energy, because you would not get enough of a spread in uniform dose to cover your tumor. So but you can modulate the beam and get this spread out Bragg peak, and we see that gives you a better dosimetric distribution than X-ray beams would give you. And so the question was, but we measure dose, but we don't account for the well, actually we do account for the radiobiological effect, but right now with proton therapy, it's done in an averaged way. We just assume that the RBE, the relative biological effectiveness of protons across this whole spread out Bragg peak where we're treating the tumor is a constant, in that it's 10%. However, in reality, we know that it's going to be less than 10% near the entrance and higher near the distal side here, where it could be really close here. It could be up to, almost a factor of 2 higher, but typically around 30% higher around this region. And you can imagine if your setup of the patient is slightly off, you might be hitting a critical structure here. And if your because your RBE is much higher than you think it is, the relative biological effects than what you're accounting for. And so it would be nice to have, again, a DNA-based detector in certain clinical situations before a patient is treated, to put it in the beam and actually make sure that for that particular treatment setup, the patient is not going to get a more damaging effect than you expect. So we designed this setup for the experiment where we have what's called solid water. It's basically these tissue equivalent blocks to mimic, a patient setup. And we put these blocks, we stack them. And here, the darker, thinner blocks here, that's where we put our little DNA detector in the middle. So each one of these blocks, we put a little DNA detector at the center of the beam. And we wanted to measure the relative biological effectiveness at different points. So if the beam is coming in this way, you would get a depth dose deposition curve looking like this. This is the red dots and the blue dots here represent just two different field sizes. But as you saw before, you get this plateau region. And so we put these blocks, our detectors in these blocks, so that we can get the measurements at P1, P2, P3, and P4, different depths. And here the dose is supposed to be constant. However, we know the relative biological effectiveness will not be the same. And so we wanted to measure the relative biological effectiveness using our DNA-based detector at these different depths. And then measure that relative biological effectiveness by exposing that detector to 6 MV X-ray radiation, which is typically what could be used an external beam now, because, like I said, proton therapy is on the rise, but still most radiotherapy treatments are done using megavoltage X-ray beams. And so we did this experiment. It was a preliminary run, and we got a couple of gels. They look like this. And so we see that the control is like we expect. Most of the DNA is supercoiled. Of course, it comes to us in about 90% supercoiled. So it wasn't, it's exactly what we expect. There was some open circular form. The LINAC, here is the DNA that represents the irradiation from a LINAC beam. And we see, there's the supercoiled has now turned into open circular because there's a lot of single strand breaks. And there's a few double strand breaks as shown by the linear band here. And at those points in the proton beam, we see in the entrance of the beam, it's pretty faint, not as many double strand breaks as when you go into the plateau region. The double strand breaks gets brighter. And so we got what we kind of expected. However, we didn't feel we got enough samples in that first preliminary run that we did to have us to just quite yet publish these results. And then COVID-19 hit right about that time. And we haven't been able to go back to that Proton Center. It was Loma Linda. We were collaborating with Dr. Andrew Rowe at Loma Linda, and they got hit pretty hard. He was, there was some, he had a baby, so he couldn't go in to work as much, and they weren't allowed to let me or my student go on site. We weren't allowed to go on site because of the COVID-19 situation, and we still haven't been allowed yet. So this study's been put on hold for now, but hopefully in a few months, I'm being hopeful that we'll be able to go back there and get the more results and conclusively show this and have RBE measurements for proton beams. So to conclude, we've constructed a DNA-based detector to measure DNA damage from radiotherapy treatment beams. We've used this for our research studies right now. And however, my goal ultimately is that we can build this so that a clinical medical physicist can also use it and use it to do quality control on pre-treatment plans using heavy particle beams. And because sometimes the doctors want to know, are we safe here? Or and right now the treatment plans tell us the dose, but they don't tell us the relative biological effect. And at some points, for certain patients, we need to know if that relative biological effect could affect the treatment, because there's a critical structure there, and motion uncertainty in the setup could affect the delivery in such a way that the patient may end up having an adverse effect to the critical organ. So we also found that the presence of a 1.5 Tesla static magnetic field during radiation delivery does not increase DNA damage. And so we expected it wouldn't increase cell kill. And so it's safe to deliver the radiation with the static magnetic field there. And we're planning for more measurements of the proton relative biological effectiveness across the spiral bright peak. It's in progress. Like I said, we hit a snag with COVID-19, but we're hoping to go back to that. So with that, I want to acknowledge Dr. Andrew Rowe, our collaborator for the proton beam irradiations at Loma Linda University. Dr. George Madrosian, we did those measurements with high dose rate brachytherapy unit, and he's at clinical physicist at Genesis Healthcare Partners, and Dr. Jordy Pang, who we collaborated with on the theory of that MR, static magnetic field damage, and a couple of former students who have worked on those magnetic field measurements, Lonnie Fuller and Erica Brescia, who are now actually currently in our residency program. And finally, I just want to thank you for this opportunity to give this colloquium here, and I'll open it up for questions.
Okay, thanks a lot. And so yeah, now.
Sorry, my speaker went off, so I don't know if anybody had any questions.
Okay, yeah. So if anyone has any questions, go ahead.
Hey, Mauro. It was a good
Hi.
talk. About 15 years ago, I listened to a talk by Dr. Ruth Wilkins from Health Canada. And at the time, I think anyway, they had some funding from the government about, dirty bomb. And she was looking at, I think she was looking at blood. So she was looking at DNA damage in the same, looking at the double strains. And I think she was using some microscopy techniques. I was
Yeah,
wondering, have you looked at that and maybe
yeah.
optical methods?
So I'm sure what she was probably using was that assay I mentioned in this talk. It's called the gamma H2AX, H2AH assay. It's basically when DNA damage is done to cells. So what happens is if you get exposed, say from an accident, the lymphocytes, the circulating lymphocytes in the blood, they'll undergo DNA damage, and when they undergo that damage, the cells try to repair. Well, they'll first detect the damage, so they'll recruit these proteins to the site of the damage, and they're the histones. So the DNA wraps itself around these proteins called histones, and they accumulate at the site. And there's a fluorescent, biomarker for those histones, and that's that assay there. So what she was probably doing is adding that histone biomarker to see where it would light up. And then, so what they would do is then count the number of foci, which represent the number of double-strand breaks to the cells. So you could do that with a really small blood sample. And actually, this is another area of interest I have, and another direction I want to go next, and take it into radiotherapy. So another project I wanted to do is where they had to draw a lot of blood to do that before. Well, not a lot, but a tube of milliliter level blood to then analyze the amount of DNA damage in the lymphocytes, and then they can correlate that to how much dose the person was exposed to. Say if it was a dirty bomb or whatever, or even if a nuclear reactor explodes or there's some kind of accident. But now I think there's a way to do it where if any of you are familiar with the just a pinprick when you're trying to get a glucose measurement of your blood with a small, really tiny amount of blood. And what I wanted to do is to, one of my projects I wanted to do is to do that assay with a really tiny amount and apply it to radiation therapy. So then we can, for example, in radiation therapy, there's been a lot of other developments, like something called ultra high dose radiation therapy. It's called flash therapy, which I think would cause a lot less damage to lymphocytes because the delivery is over. You deliver the whole treatment in 1 to max 3 fractions, so big dose in very few fractions and very little time, less than a second, and so you're not going to hit that many peripheral cells, those cells flowing in the body. And so if you did that, you can show one of the advantages, just to clearly show that, of flash would be that you're going to do a lot less damage to these kind of cells. But you can also use it for space travel. We know that people in space are going to be exposed to protons and other ionizing radiations, and then you can quantify that damage by just taking a little tiny bit of blood, like you would if you were doing a glucose test, and not have to draw a lot of blood and then put it on there in amount, just measure that chromosomal damage. Sorry, that was a long answer to probably a short question.
Yeah, I was wondering about this, optical microscopy, because like people in biophysics, they use them a lot. And yeah, I don't know a lot about it, but I know that they use all these techniques, fluorescence, and I wonder if we could do that too, like be more physical method instead of the wet lab, like you said.
Yeah, and that's what I'm trying to do with those 4 techniques that I was mentioning. Yeah. Get it. So yeah, because physicists are not going to measure that. They're not going to adopt the, So my plan and my hope is that one day we'll have, these little, I don't know where, little detectors like this that you can put in the beam, and then easily within at a clinic just get the pipette the DNA out, put it in your detector, and get a readout without having to make gels and do all this other wet lab stuff.
Yeah. Good.
I've got a couple of questions.
Sure.
The first one, and probably the more complicated one, is when you get your DNA samples and they're 10% in the circular configuration, there must be some chance that a single strand break on one of those could open it up into a linear configuration. And do you know have a handle on what chance that is, and is that something that you need to adjust for in your data?
Yeah, it's a good question. Yeah, you're right. Yeah, they will. So this is why when we run it, we always run a control at 0. We actually ran in this experiment 30 of these, so we can quantify it exactly how many of these, because these circular are going to change to these. But if we know how much circular is there to begin with, we can account for this, and that's why we also did this model. Whatever is it? Yeah, we had that graph. Yeah. So here we start here. We had actually started with quite a few of those circular conformations, and with radiation, we see this really rapidly rises. And we know some of those, but we can account for it here because we know the initial yield of it, so we can adjust for that.
Okay.
Yeah, with the dose.
My second question. Well, thank you for that answer, and it's something I was wondering. And the second one is just with a 1.5 Tesla MRI magnet, how does that compare to the strength, the field strength in the bending magnets? Just the magnets that you have to use to steer the proton beam.
All right. Actually, I don't know what the. You're right. There's bending magnets in the LINAC. I don't know offhand what the magnetic field strength of those magnets is. Does anybody over there know? And just for background, those bending magnets are there because you're accelerating these electrons, and you have a transmission target. So unless your accelerating structure is pointed directly at the patient, which with the higher energies it's not. So you want to change the path of the electrons so they circle around and come through the transmission target so they can hit your patient. But yeah, that's a good question.
I don't offhand know what the strength of the bending magnets is for a LINAC like that. I was also thinking of the proton accelerator at Loma Linda, because I've seen that. I saw it 24 years ago, I think, before they started it up.
Right.
But there's for those of you who haven't seen it, the gantry on which those bending magnets sit is the size of a house, literally. and they steer these things to sub-millimeter accuracy. So if they could make stronger magnets,
incredible.
they would make the whole thing stronger. But it may be more efficient since last I saw it 2.5 decades ago.
I don't think it's changed very much at Loma Linda, but they do have a proton therapy machine here in San Diego. When I actually moved to San Diego in 2012 for this job at San Diego State, I had an opportunity to see, to go to the Proton Center and see in the back, and it's like stories high. It's incredible. And you wonder how, and this is nothing compared to, the Large Hadron Collider or any of these other things going on in particle physics. But to me, I was amazed that, you know, these things are built and they can deliver to such precision.
It's amazing. Yeah.
I had a question about the magnetic field. So you got basically a negative result. Do you know if you increase the magnetic field, if that would maybe change? I think the magnetic field is too small to basically matter for electrons. If you calculate the bending, I think it's pretty minimal. So I haven't read that paper that you mentioned, the theoretical one, but
yeah. Well, what that paper showed was that it theoretically becomes relevant at energies like you would get with HDR, which are, you know, the Iridium 192, which are much lower energies than the LINAC. But you're right. So it's a combination of the magnetic field strength and the radiation energies, basically the electron velocity, because, you know, the electrons that are set in motion, which deposit the dose, are going to be at a much lower velocity at those lower energies than they are at the megavoltage energies. And it's there, you know, for HDR, they travel less than a millimeter. So they're not going very far at all compared to a LINAC where, you know, they're on the order of centimeters in terms of the travel. For example, 6 MV beam would travel like on average 3 centimeter would be the range. So yeah, you're right. And the reason we did the study with 1.5 Tesla is because that's the highest clinical magnetic field that's used in the machines, as we see here. And the 1.5 Tesla MRI, you know, that's also been a standard magnetic field strength used for imaging, you know, in MRI units. Now a lot of them are 3 Tesla, but we don't have 3 Tesla, you know, incorporated into a LINAC system like this yet. And of course, there's the practical reason is you could then, so there's not just a radiobiological effect, but a dose effect. We know that even at 1.5 Tesla, even though there's no radiobiological increase, you do see increases at interfaces. So for example, going from normal tissue, like water equivalent type tissue to like lung or something like that, the electrons will, when they go to that lower density tissue, will actually swirl backward, cause an increase in dose upstream. So there are differences in dosimetry in some of the spots.
Yeah.
But so that's why I think ViewRay here, they used cobalt 60 so they wouldn't have to deal with, you know, these machines are allergic to each other. They wouldn't have to overcome engineering difficulties. And they also use the smaller magnetic field strength, I guess, maybe to make that dosimetry a little bit better.
Yeah.
I don't know for sure though.
Yeah, I've seen a talk. They did an interesting thing how to avoid. If you put actually rings of metals, the magnetic field cancels. So it's actually very interesting. It's electrodynamics. So you can calculate that if you actually put a bunch of rings, metallic rings, concentric in the middle, you can actually run basically a wire without being affected, or electron without being affected by the magnetic field. So they found interesting solutions to that. Yeah, it's pretty neat.
Wow.
Yeah, I've seen a talk, I think, 2 years ago or something.
So I think I see, Yizzy, did you have a question?
Yes, so I think this is a great talk. I'm pretty new to this field, but I was able to follow the logic. So I understand now there are 3 different forms of DNA, and then they form different bands when you use the gel method. So my question is, within each band, for example, the band for the open circular shaped DNA, does each DNA, they have exact the same property, or they have some quantitative difference within each band, or is there any way you can distinguish the difference? Or maybe alternatively, I can ask the question this way. So before the single strand breaking or double strand breaking happen, as you increase the dose, is there anything happen to the DNA molecule themselves before the breaking really occur?
I don't know. From what I've known from, and this is just from the literature that I studied, is when you do a single strand break, from what I understand, is supercoiled will just become open circular. I don't know the exact underlying physics or why that happens, but for some reason, if you nick one of the strands, I don't know. I looked for that myself. What is the underlying physics for this? I don't know why, but it happens. It ends up unraveling that whole supercoiled form into a circle, and then if you nick it again, or if you go double strand break from the beginning, you get the linear. Now the linear one is easier to understand. You break the bands here. These are negative. These are just going to repel, and I guess it has to do somewhat with that as well. These are going to want to be apart if they don't have to be together. But deeper than that, I can't give you an answer. I'm sorry.
Thank you.
Oh,
Yeah.
I could add a comment to that, and this is based on teaching radiobiology, but it was 20 years ago.
Okay.
So any updates to this are welcome. Just when you're talking about biological effects, the main effect that they always look at is breaking DNA strands. And one of the reasons for that is that the cells have a repair mechanism, because DNA gets damaged all the time. If you get even a single strand break, that can be fixed inside the cell. And also any smaller damage like that, like just knocking an oxygen off or something like that, DNA does fix itself, and I don't know very much about that, but it's the breaks that they really look for, and a single strand break can get fixed. A double strand break usually cannot be fixed, and that's what leads to mutations. And that'll affect either genetic mutations that cause birth defects, and also starting to cause cancer, or just outright killing the cell. They're all related. All of those are related to the double strand breaks.
Right. Yeah, that's right. And that's what I was saying here. Yeah, in the lecture as well. It's the double strand breaks that we care most about. Yeah, and it's for that reason. Yeah, the cells have the most difficulty repairing the double strand breaks.
So are there any other questions, comments?
I have two questions that you may consider very elementary.
Okay.
First is, a massive radiation dose, such as in excess of 6,000 rems, can be instantly fatal to a human being. Does the disruption of DNA cause this, or is there some other cause?
It begins with this, yeah, the DNA double strand breaks. And yeah, a single dose that high to the whole body, certain cells would because of the double strand breaks, lead to cell death. Certain cells would go first. You have early responding and late responding tissues. And so again, in radiobiology, they can triage patients that get exposed to a single large dose based on the symptoms. So the sooner the onset of those symptoms are, the worse and higher the dose was probably there.
Also, in a massive dose, sometimes you see lesions. What are those caused by? Lesions on the exterior of the skin. What causes that?
I would say, well, skin is one of the early responding tissues. So if you see that, you wouldn't see it right away, though. I think you probably would see it after maybe a few days or a few weeks. But that would be the cell, the damage caused, the skin cells, when they're going to, you cause a whole bunch, they go necrotic and there's a difference between necrosis and apoptosis. So they would commit cell suicide and they would be regenerating, but it's like the sunburn, I guess, right? Where you damage the skin and then it peels off and you get, but
Yeah.
I think that's not one of the symptoms you look for. If you see that early on, you're in big trouble. It's not, and it's not like in the movies. Every time I see these science fiction movies where they're exposed to these high doses of radiation, and you see these, real-time skin going necrotic and stuff. It's not. That would have to be huge doses.
Well, they told us in Navy Nuclear Power School that unlike the science fiction movies, no human being has ever been so radioactive as to glow in the dark, not even a dead one.
No.
But the thing is, the first fatalities at the Idaho, now the Idaho National Laboratory, where I did my prototype work for Navy Nuclear Power, the first ever fatalities due to a nuclear accident at this facility, there were reportedly lesions immediately appearing on the skin of the technicians in the accident, which it was of EBR-1, the very first ever breeder reactor. And they must have gotten a massive dose, which is quite possible.
Right.
The other question that I had was, the DNA in your DNA radiation detector, is that completely synthetic DNA? And if not, what organism does it come from?
Oh, yeah, it's not synthetic. It comes from E. coli bacteria.
Okay.
It's the plasmid DNA. So plasmid DNA from bacteria is actually the DNA part that is usually supercoiled. It is the DNA that actually protects bacteria from antibiotics. That's how they gain antibiotic resistance is from that part of the bacteria. It's not so the bacteria have the DNA that it uses to replicate, and then it has this plasmid DNA inside it as well, and that's the part that it comes in these kind of forms, and also gives it the advantage of quickly overcoming, evolving to have resistance. So some bacteria are not, would have that resistance from the plasmid, and then it would get
using DNA would probably require quite a bit more paperwork than anybody would want to do without a good reason.
Oh, well, this DNA here is, you can just buy it. There's no, I don't need any IRB approval for this, because we're not changing the DNA. We're not doing anything to it. We're just using it. We're just irradiating it. So it's actually, I can ship this to, across the country and without having to get special paperwork or anything for it.
Okay. Well, great. Thanks for a great talk.
Yeah, you're welcome. Thanks again for the invitation.
All right.
Yes, thank you. I just have a comment about Fred's comment about massive sudden doses of radiation. Many people do not realize that in the US development of atomic weapons, there was a third atomic bomb. It was called the Demon Core. And if you look up online, mistakes made with the Demon Core killed people. Cherenkov radiation, for example, happening all of a sudden because somebody dropped a tool. So there you go. Just, you can look that up on your own. I just wanted to make that comment.
Yeah, it was in the Pacific. Yeah.
Oh, cool. Thanks. So are there any other comments?
Yeah, I have a question.
Okay. Go ahead,
So
guys.
in the graph of the depth versus the relative dose, why is it that the proton beam and the carbon ion beam, why do they drop in relative dose when as the depth increases?
You mean this drop back here?
Yeah, yeah.
They're slowing down. So these are charged particles. So as a charged particle slows down, it loses more and more of its kinetic energy in the form of it ends up transferring that energy to ionize the atoms in the material it's traveling through. And in the beginning, that doesn't happen so much because they have a very high velocity, very high kinetic energy. And at a very high kinetic energy, this is something called stopping powers. So if you take a course in radiological physics, you'll learn about this thing called stopping powers. And it's about how much energy a charged particle loses as it's traveling through some medium. And so if it's got a high velocity, it turns out it loses very little energy. As it slows down, it starts to lose more and more and more of its energy. And near the end of its track, it just deposits most of that energy near the end of the track, and then can come to a complete stop. And that's why you get that complete tissue sparing. Whereas X-rays, they attenuate in the material. And the reason it increases for megavoltage X-rays is because there's what's called a buildup region. So basically electrons, the X-rays are indirectly ionizing. So they have to set a charged particle into motion. So they set charged particles, and these are electrons in the motion. And as the electrons begin to get set into motion near the surface, they'll build up and build up until there's a quasi-charged particle equilibrium, and that maximizes at some point, and you get this partial charged particle equilibrium at depth here. But the X-ray beam attenuates exponentially, never goes to zero. So you'll still end up getting dose even after you may not want it anymore, because there's still X-rays setting new charged particles into motion and going downstream. Whereas if you have a charged particle beam, it's that property of charged particle beams. They'll slow down and then deposit most of their dose near the end of their track. And that's because they're undergoing the Coulomb force. The reason they're slowing down is every time they're going past an atom, there's the interaction mechanism is the Coulomb force field between the electrons or the nucleus and the charged particle. And that Coulomb field, if the charged particle is traveling slower, it's going to be in that Coulomb field for a lot longer. And so, of course, and this is just an intuitive way to see it, that if you have a charged particle zipping by really fast, it's going to barely feel that Coulomb force field. Whereas as it slows down, it'll feel it more and more and more. And then it'll start to, that Coulomb force field is going to affect it more and more, and it's going to slow it down more and more as it loses energy by ionizing and exciting the atoms. I hope that clarifies.
Yeah, it does. Thank you. Thank you very much.
Welcome.
Okay, so if there are no other questions, let's thank Dr. Tambasco again for a great talk. And Mauro, thanks again for a great talk. I'm going to stop the recording.